Hi, everyone. Welcome to QCBio seminar series. It's my pleasure to introduce Ariel Wu. So Ariel is a PhD student in computer science. She is co-advised by Elazar Eskin and myself. And Ariel is broadly interested in computational methods to understand the genetic basis of diseases. And today she'll tell us about some recent work on how to estimate genetic correlation from extremely large data sets. Ariel, are you able to share your screen?
Yeah.
Okay, take it away.
So hi everyone, I'm Ariel. So today I'm gonna share our work on estimation of genetic correlation for biobank-scale data. So before we jump into the details of the method, let me first talk about the motivation of this project. So in general, in bioinformatics, a fundamental question people are trying to understand is how genetics affects the phenotypes such as height and BMI. So in this, here we have a toy example. Assume that we are interested in two traits. One is BMI and one is height. And then just to make the toy example simpler to understand, so we assume we have 4 genetic variants and we have 2 samples have the observations on these 2 traits. So, and then for each phenotype, we try to estimate the effect sizes on this 4 genetic variants. So we have the red ones are the effect sizes on BMI, and the green ones are the effect sizes on height. So we see that the first 2 variants have both high effect sizes on both traits. So it seems like they are highly correlated. And then for the other two genetic variants, SNP3 and SNP4, it seems like they have different effect sizes. So it seems like they are not correlated. So we know that BMI and height are correlated because BMI is dependent on the inverse of height. So the question we are trying to understand here is that what is the genetic basis of this relationship? So in order to quantify this, people define this genetic correlation. So rho G is the proportion of correlation of a pair of phenotypes explained by the correlation of the effect sizes of the genetic variants. So to make this definition clear, so we have the correlation between the effect sizes of BMI and height. So it's the correlation between these 4x1 vectors. And then we have the correlation of 0.79. And also we have the correlation between 2 traits, BMI and height. And then we know they are inversely dependent on each other. So the correlation is -1. So the genetic correlation by definition is the correlation between the effect sizes divided by the correlation between the traits. So it's -0.79. So so far we have defined genetic correlation, and then so the question we need to ask it now is that how do we find the effect sizes of genetic variants? So the answer is that we use the linear mixed model. So we start with a simpler model. So we start with the linear model with 1 trait first. So we have, assume we have the genotype, we have n individuals, and then we have observations on M genetic variants or M SNPs. And then we have vector, n by 1 vector of phenotype, which is Y1. And then we define, or we assume the generative model of the phenotype. So Y1 is given the effect sizes and the genotype, it's the linear combination of the genotype plus some noise. So beta 1 is the vector, is a n by 1 vector, which is the effect sizes of each genetic variance for this phenotype. And then epsilon 1 is the residual effect. So typically in most of cases, we have more genetic variance than the number of samples. So that's why usually we assume that the effect sizes is coming from a normal distribution, which is zero mean with some variance, sigma G1 squared. And then this, with this assumption, we have, this is what we call linear mixed model. And then now let's proceed to the bivariate linear model. So now we have two phenotypes. So similarly, for the second phenotype, we also have this generative model, where it's also a linear combination of the genetic variance and some noise. So here we have another vector beta 2, which is the effect sizes of the genetic variance. And then how do we relate the genetic effects? So we assume that for each genetic variance, the effect sizes for two phenotypes are coming from a multivariate normal distribution, where on the diagonal term are the genetic components divided by the number of SNPs. And then the off-diagonal terms are is defined as genetic covariance, which is gamma G. And then by our definition, the genetic correlation rho G is equal to this genetic covariance gamma G divided by the square root of the genetic variance components. So now, just a recap on linear model, bivariate linear model. So we have the input data, which are two vectors containing the measurements on two phenotypes. And we also have a genetic genotype matrix X. And then the goal is to, or the parameters we're trying to estimate are the variance components, sigma G1 squared, sigma G2 squared, and the sigma E1 squared and sigma E2 squared are the environmental components. And then we are also trying to estimate the genetic covariance gamma G, and then having these estimates, we have an estimator for genetic correlation, which is rho G. So one set of the common methods are trying to estimate this is likelihood-based method. So in general, these are iterative optimization algorithm, and then it solves the computation burdens come from solving the inverse of n by n matrix in each iteration. So this method is most it's the most accurate method, since it's using all the information and then solving the likelihood, but it's very computationally expensive. So the runtime is between quadratic to cubic to the number of samples. And then the state of art software is more than 10 days to run on biobank data, because right now, so for biobank, we have 500,000 individuals and 500,000 variants for array data, and then we have more for imputed genotypes. So this method, it's very computationally expensive. So the goal of our method, the goal of our project is to trying to come up with an estimator of genetic correlation, which is efficient to run a larger data set. So in our method, we use this method of moment method. So the high-level idea is that this method of moment is in general trying to minimize the distance between the population moments to sample moments of y. So in our method, we use the first two moments. So the first moment, since we standardize the phenotype, the first moment is the expectation of y, the expectation of phenotype, which is equal to 0. So what we are trying to do is to solving by minimizing the sample moments, which is the covariance of y, to the population moments, which is y transpose. And then after we solve this equation, we see that we have a closed form solution for genetic covariance gamma g, which is on this equation. So we see there's a term K here, which is the kinship matrix, which requires the multiplication of x transpose. And then, so the idea here is that the computation of kinship matrix is also very expensive. So we use we use an estimator for this trace of k squared. There's this identity where if we have a symmetric matrix, we can use some random vectors where the expectation of random vectors transpose matrix A random vector is equal to the trace of this matrix. So we use this identity, and then we have an estimator for trace of k squared. So this is the key idea of this method to make it computationally efficient. So so now we have to sum it up. So we have the closed form solution for genetic covariance, and then we, in order to solve this computation bottleneck of trace of k squared, we use an estimator of this. So then we have an estimator for trace of k squared, and then we have a estimator for genetic covariance. And then having the genetic covariance, we can we use the previous definition to, and then we reach the estimator for genetic correlation. So we have described our method, and then here are some properties of our method. So firstly, our method is computationally efficient. So we measure this using taking subsets of UK Biobank, and then we run methods. So there's a typo here. So our method is called score, and then this pink, and then this blue lines are both score, where score overlap is a shortcut version of our method where two phenotypes have exactly the same measurements on the genotype. So we see that, and then GCTA GREMO is solving is a maximum likelihood method, which we described earlier. So this is the most accurate method. And then we have GCTA also have a version of method of moment version, which is called GCTA HE, but it's not using the estimator for trace of k squared. So this is where the computation burden is coming from. And we see our method could run on the full biobank data within 2 hours. And then we try to estimate the estimation accuracy for our method. So we compare to 2 individual level database method, GCTA GREMO and GCTA HE that we saw earlier. And also we compare to 2 other methods, HDL and LD score. These 2 methods are summary stats based method. So the good thing about summary stats method is that summary stats is it can be considered as a function of the effect sizes. So we could directly use summary stats to estimate without using the genotype data. So it preserved the privacy, but at the same time, the drawback for summary stats-based method is that first it requires a reference panel. So if there's a mismatch between the true sample and then the reference panel, then the estimation could be biased. And the other thing is, since it's summary stats based, so in general, it's less statistical efficient. In other words, it has a bigger standard error, so it tends to which we will see later in the real application, is that it fails to identify some of the significant correlations. So in here, we consider different situations with different true. So we perform simulations taking a subset of UK Biobank, and then we simulate a pair of phenotypes, and then we consider different architectures. For example, we consider infinitesimal model, which is the model we described earlier, where each genetic variance has a small effect on the phenotype. And then we also consider the non-infinitesimal model, where only a subset of where genetic variants have effects on the phenotype, and the other ones are the effect sizes are 0. And we also consider different heritability or different values of genetic components. And then we consider different value of the true genetic correlation. And then, so in total, we have 48 genetic architecture. So if you're interested in the technical details of these simulations, you could take a look on the preprint on bioRxiv of our paper. So here we just show the summary of these 48 architecture. And then we see that the likelihood-based method GCTA-GRAMO, we consider this one as the gold standard of the estimates. And then we plot the mean square error, and we see that our method score is almost as good as the likelihood-based method. And then we see HDL and LD score, where these two summary stats-based method, they tend to have tend to be less accurate than individual level based methods. And then we want to assess the statistical efficiency for this for these methods. So we plot the relative standard error between the methods versus the likelihood-based method, GCTA-GRAMO. And then we see that so our method score is having a small standard error, so it's very close to likelihood-based method actually. And then we see summary stats method, HDL and LD score here, especially at LD score. They have very big standard errors compared to our methods and the likelihood-based method. And then, so having assessed the property of our method, we show that it's computationally efficient, it's statistically efficient, and then also the estimates are accurate. So we run our methods on traits from UK Biobank. So there are some measurements on the biometric measurements, and also some diseases and some physical conditions such as sleep duration, get up time. And then in the top left triangle, we plot the estimates of LD score, which is the most popular methods right now. And then we, on the right bottom triangle is the estimates of our method. So we see in general, these two methods reach similar point estimates, but we mark the some pairs with stars. These are the ones where our methods found it's significantly correlated, where the other method didn't because of the standard error. So, so in summary, our method is efficient and it's also as accurate as likelihood methods. And then, so for larger SNPs and individuals, usually a small number of random vectors should be adequate because our method depends on the number, the runtime depends on the number of random vectors. So we did test the number of random vectors with being 10 and 100, and then we see that using only 10 random vectors is pretty adequate. So in the real analysis we see earlier, it's actually using 10 random vectors. And then it could handle the case where we have traits with partially overlapped samples. So these are the cases where, for example, one of the traits could be, say, a disease. So most of the sample doesn't have observations. And the other ones, other ones could be some measurements like BMI or height. So these two traits could be partially overlap. And then this method could solve this case. So for future direction of this method, so one interesting question people might want to understand is that, is this genetic correlation the same along the whole genome, or there's some difference or enrichment in certain regions? For example, we know that in genome-wide association studies, we see some of the disease they have, or some of the traits, they are very, very polygenetic. For example, for heights, there are tons of associated, significantly associated genetic variants. And for some of the traits, there are only a few associated, significant associated variants. So we want to understand also for genetic correlation, is that the same thing for different pairs? So for example, we show an example where this is the genetic correlation between the alcohol intake and the BMI. So we have measured the genome-wide genetic correlation is being 0.38. And then we further partition the genome into different groups. So this is partitioned by the SG annotation, the specific gene expression annotations. And then these groups are further divided into 9 groups where they are describing different functions. And then it's coded in different colors. And then we see in this specific case, it's pretty, the genetic correlation within each group is very similar. But in some of the pairs, we might expect to see different genetic correlations in different annotation. So this is a interesting future direction. So any questions?